Go.
Hope it dies.
You guys should be able to do 2 things for me. I have a DNA molecule up above that I've actually already unzipped. I've placed letters representing the bases that are along this DNA molecule. You guys should be able to take that code, that DNA code, and first tell me what the complementary DNA code would be, like as if this DNA molecule was undergoing DNA replication. Then you need to totally, on a separate process, take that same code and make it undergo transcription to make whatever molecule. You guys should already know what's made as a result of transcription. So you'll basically fill in the question marks here. So you'll also give me the code that this would transcribe to. See if you can do that.
I'm confused.
I could do the second one, not the first one. I could be confused.
You could do both of them.
You confused the crap out of me.
No.
Yeah,
The top
you
one
really
here.
did.
Let me just kind of give everybody a few seconds, and
You want us to
I'll
fill
give
in
you more hints.
both sides of it, right?
Nope.
Yes.
Ignore the 5 prime 3 prime. Actually, that's not. If that's the only thing that's confusing you, let's take away that.
That's not the only thing that's confusing me.
Okay. You know how to do this. You are. Well, let's do it one at a time. Let's totally take away this part 2. You are taking this code and you are going to have that part undergo DNA replication. You want to make more DNA to make question mark. Your question mark would be DNA. In order to do this, number 1, you just have to tell me the complementary DNA code. All right, what always goes with T?
A.
What always goes with T?
A.
You get the idea. I have a T here because I'm still talking. We're going DNA to DNA.
Oh,
That's all I'm looking for. You just made yourself another strand of DNA. That's question 1. Question 2, though, this is what we talked about yesterday. Now you're going to take that piece of DNA, and it has a whole different use. You're taking that DNA, and now you're going to make what by transcription?
RNA.
RNA. So do the same thing, just make RNA.
U and U.
Well, A, they would still be A's.
What is?
Yep, because A's still exist.
Yeah.
G, C, there you go with the U. G, A, I know everybody gets that confused, but it's A's still exist. U. That's what I wanted you to do. The codes are not the same, depending on if you're making DNA, like in number 1, or if you're making RNA in number 2. This is still, your DNA still is used for both of these processes, but it's going to just have a different result depending on which process that portion of DNA is meant to perform. So that's all I wanted you guys to do. Okay. All right. So then today, actually, let me think another thing that we talked about yesterday. I think the last thing that we talked about yesterday was the fact that, yesterday we went over the process of transcription. You don't have to write this down. This is just review. We went over the process of transcription yesterday. Very similar to what you did in number 2 up here. And remember, the whole point of that, it was basically part 1 of our 2-part process to make protein, but transcription is just going from DNA to RNA. That's what we did yesterday. Then the last thing we talked about was the fact that in the end, you're going to have this messenger RNA molecule here, but some parts aren't going to make the cut. Some parts are going to get scissored out. Which are the name of the parts that are going to get the axe, that are going to get introns? Very good. So we're going to see this messenger RNA molecule just get chopped up. And let's say this, if it's not an intron, remember the introns are going to get X'd out. What are the name of the things that are remaining behind?
Extra.
The exons. So it's the exons that are going to get literally cut out, glued back together, and you'll just have a better and smaller little messenger RNA transcript there. It's ready to roll now. Now we're ready to complete the second part of this process, which is called translation. So this is new stuff here. Translation now is going to go from the RNA, and in particular, you can put messenger RNA.
You said we didn't.
Yep, this is new. Yeah, you got to write this down.
Oh, this.
Yep. You're going now from messenger RNA and to the ultimate final product, which is whatever protein you want to make. Remember, guys, I mean, this is going on inside of you right now. You guys, in order to stay alive, have better be generating lots of different kinds of proteins. Your DNA is providing the ultimate blueprint to make all those proteins. All right, so let's look at part 2 of this process. It's a little more complicated. What happens here, and this is going to be hard to draw. I might show you a picture first. That might be easier. Let's say I had a messenger RNA code that had the following sequence. Okay, I know that's a lot of letters, but bear with me, there's a point to this. So I have just drawn a small portion of a messenger RNA molecule. That definitely has a code in it. At this point, it's important to break the code up into 3 letters each. Each of these groupings of 3 letters is going to be referred to a codon. So codons are always part of the messenger RNA molecule. Here's the deal. Each codon will go on to code for 1 amino acid. Remember, guys, it's going to be a bunch of amino acids linked together, so each amino acid is a circle. We're going to build ourselves up a protein 1 amino acid at a time. Each of these 3-letter codes is going to code for the 1 amino acid, and they're going to follow in the same order. 1 amino acid at a time. Here's where people find this kind of fun, because it's going to be you guys who need to figure out what each codon codes for. In other words, what amino acid results from AUG. It's at this point you guys need to pull out this one sheet of paper that you got about a week ago. Hopefully you still have it. It's that chart. Actually, you're going to need both of them. You're going to need the...
Do you have yours?
Yep. Oh yeah, you're going to need this one immediately. This one right here. This is where this thing comes in handy. It's like a deciphering code tool. And then you're going to eventually need to have the one sheet you worked on in class, so keep that handy.
I did that on a separate sheet.
Oh, cool. That's fine. That's great. That's great. So eventually, but this is what I want you guys to have right now, this sheet. I want you guys, I think from last week, you guys know how to use this thing. That's the nice thing. You guys should be able to take a codon, like AUG, which is on an RNA molecule, and from this sheet of paper, you can tell me what amino acid goes along with that code. So all these big words in here are amino acids, is what they are. So AUG, someone tell me, what does AUG go on to code for? What amino acid? Start, which is also, and it is the start codon, which is also called methionine. I'm just going to write the word, I don't want to write meth there, I'm going to put methionine, and I'm going to put start. All right, take the next one.
See, I don't understand.
You don't know how to do this?
I don't get this.
It's, actually, raise your hand. Let me just kind of briefly go over how you use this sheet again. You have 3 letters here, A-U-G. You start at the left-hand side. You see a U, you see a C, you see an A, U, a G. The first letter is an A, so go down to the A, which is kind of the 3rd letter down, and kind of put your finger there for now. The next letter is a U. For the second letter, you go along the top and find the U. You'll see the U is in the first category. Put your finger there.
Oh, I get it.
Now G. You gotta go to the right-hand side. Where the 3 of those letters meet up and hook up in the middle, that's where you put the amino acid. That's what the amino acid is. So see if you guys can do the remaining 4 here real quick. I think you guys can figure out the 4 other ones associated with this. So GGC. You get the idea. To abbreviate here in the end, guys, because I'm running out of room. All right, starts with, oh, how are we doing? Is everybody getting this?
Yeah, okay.
Starts with methionine, then you should have glycine. Then you should have isoleucine. Then you should have asparagine. I know they're big words. You don't need to memorize the words or anything. Then you have glutamine, I believe. Glutamine. You guys just need to know how to use this chart kind of a deal. Like again, remember the big picture. What this thing's doing. This chart is so useful at taking an RNA code and figuring out which amino acid goes with the code. You are building up a protein. That's all this thing is. Methionine, glycine, isoleucine, asparagine, and glutamine. I've just, it's asparagine, whatever. Okay. Did I write it wrong?
Yeah. You said,
Oh.
you said asparagine, it's aspartic acid.
Oh, okay, thank you. I looked it up wrong then. Aspartic acid, very good. Okay. Cool. Anyways, all I did was build a protein up. So remember, I mean, this is all you're doing, making proteins. Okay, now here's where there's a little more detail, but not much. I'm going to do this again because I want us to get really good at this. Let me take another messenger RNA code because I'm going to talk about a little more detail here, but let me just give you a new code. Oops, I'm going to write a T. That's bad. Okay. Hold on, let me do one more because this is just fun. All right. Here's a little more detail. Oops, I got to move this down. You guys aren't going to like me. Sorry, I'm going to move that kind of down a little bit. Remember we said before that there are these cool molecules called tRNAs. Oops, tRNAs. And that they have an amino acid on the top of them. Or maybe I didn't say this. Oh, no, I did. In your notes. Way back when I was talking about the differences, the 3 different types of RNA. Okay. This one, this particular tRNA has methionine, that amino acid at the top, and at the bottom there is something called an anticodon. And these anticodons are on the tRNA molecules. Here's the deal. The amino acid is actually going to get tacked on because this transfer RNA has an anticodon that's going to hook up with this messenger RNA codon. In this case, this anticodon on the tRNA is going to be UAC. When UAC hooks up with AUG, they are complementary to each other. That is like a match. That means methionine, bam, goes there. Okay. Different transfer RNA molecules with different amino acids on the top and with different anticodons on the bottom are going to plop down here one at a time on here, and they are the ones that are actually going to bring the amino acids to the growing protein chain. This is hard to draw. I'm actually going to show you guys a little video clip in a minute showing how this whole process works in action. But I wanted you to kind of understand where the transfer RNAs came from. Here's another detail that I left out. Not on purpose. The place where this thing is happening, the place where these tRNAs are coming in and this messenger RNA and they're hooking up, this is taking place on a ribosome. So basically, this messenger RNA is hooking up to part of the ribosome. It's just basically creating like a platform in a way, a spot. Then that's going to allow these tRNAs to come on in and do their job. Yeah.
So it connects because A goes to U.
Correct. See how they're complementary? Yep. Well, let me see if you guys, this would be, this next tRNA that comes in, what's going to have to be the code on the other end of this down here? G, G, U. That's right. I know this is like where a lot of little coding comes in, but the only tRNA that will be able to plop its butt down here on this second codon will be GGU. And when it does that, it's going to bring along another amino acid. Let me see which one in particular. GGU, glycine. Glycine is going to be the amino acid that one brings. Oh, no, wait a minute. CCA. Bad me. Bad me. Bad me. I got to look at CCA. Proline. Thank you. I heard it. This tRNA is going to bring proline along with it, and proline and methionine are going to hook up together. And it's just going to keep going down the line. Somebody figure out UCG. What's that going to code for? What is it? Serine. So the next tRNA that's going to plop its butt down here is going to have serine on top. GAG, what do we got there? Good. Glutamate. Glutamate will be the next amino acid. And then finally, what one? Stop. And I did that on purpose, guys, because you can't make a protein run on forever. You got to have some signal that's going to stop the process and cut the ties and send that protein on its way. And that's what the stop codons are for. You got to have a start codon and a stop codon. I just mentioned the stop one, and you guys even mentioned the start codon will always be AUG no matter what protein you're making. Always, always, always. The stop codon, if you look closely on this sheet of paper, there's actually 3 different codes that can code for a stop. It just depends on which one. It doesn't matter. If you get any of them, you're going to stop the process. All right, this might still be a little fuzzy. Let me show you guys something. Let me show you a little video clip. They're like 2 minutes long each, so they're not They're not all that long. I haven't decided yet. I don't think so. It's still pretty early. Let's see. Oops. Computer's taking a long time to load here. No, it's not. It's really not. It can get complicated, but it's actually it's a pretty easy process.
I like it.
Yep. Actually, every year I teach this, everybody seems to like this chapter because it's different.
Yeah, because I can understand this chapter.
Yeah, it doesn't have such big, super long words. It has more, it's almost like decoding work. It's pretty easy. But that's you always learn even from grade school that your DNA is your blueprint. It's your code for who you are. So it makes sense that this chapter is going to be all about codes.
This poster's messed up. I was thinking reading it. It said took blood from this chick about to know.
Oh yeah, yes, they did. That's actually a very interesting story. Yeah.
But then
That's a whole. I do. I want to talk about that. I can't do two things at once. Oops.
are you the one making the videos of people for Christmas? The one about Mr. C was funny.
Yeah. Mr. Sin wouldn't show us. He said that he would only show us one, and he showed us the board
With Miss
all the
K in
English-
it?
Yeah, with all the
Miss
English
K and
teachers'
Mr.
names.
Bell.
You guys will see on the date before your last day before Christmas.
The 20th?
Yeah.
How many have you made?
Oh, a lot.
Where are we gonna see him?
Homeroom.
I've only seen two.
They're about
Yeah, they're funny. Those things are great. Okay, I'm going to show you two little video clips. One of them showing transcription, and the other one showing translation. So you got to have to put both of these together in your head and realize we're making a protein. So let me see. Here.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription.
We talked about this yesterday.
With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter,
We didn't talk about
pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along.
I really don't like his voice. All you're doing here, guys, that pink thing is RNA. You are making RNA from DNA. And they're just showing you the base pairs. And you guys know how to do that. Knowing your base pair rules and the fact that you don't have T's, you're separate. There are more U's. They're just showing you, and we talked about this yesterday, that's RNA polymerase. That's the main enzyme involved in this process of transcription. Changing DNA into RNA. This is step one of our process. Okay. Now you've just made yourself some RNA. Now the next step is we're going to take that RNA and we're going to make it by the process of translation. Hold on, I do want to point out one thing here though before I go on any further. Before they start moving things. Keep in mind, this is taking place in a cell. That purple thing is a nucleus. So the process of transcription, going from DNA to RNA, takes place in the nucleus. That RNA is going to have to kind of slip out of the nucleus, and the second process of translation occurs in the cytoplasm. And in particular, you're going to see it take place on a ribosome. So let's get rid of his voice and I'll talk with it along the way. So this is translation. You have your messenger RNA, the pink thing, and the yellow thing. So your pink thing is a messenger RNA. You have your start codon, AUG. I like this video because it's going to show these little transfer RNAs come on in for a landing. The transfer RNAs, oh, that's the ribosome, first of all. That's the one unit of the ribosome. It's like a platform where this is all taking place. This is why ribosomes are needed. That thing's a transfer RNA. It's green. It looks a little different. It doesn't look like a T, but see at the very top, it's holding on an amino acid. That anticodon is going to hook up with that codon, and bam, it's going to plop itself into position. You'll see another tRNA enter the picture. Oh, first the top part of the ribosome comes into place, and they go into some detail that I'm not concerned that you guys know about. Just that brown thing is a ribosome. These things are all over the cell. There's another transfer RNA coming in with another amino acid on top. Its anticodon's going to hook up with the codon. Those two are going to hook together. Those are two amino acids at the top, and they just formed a bond between themselves. It's going to work its way down the line, bringing in yet another transfer RNA with another amino acid on top. They're going to form a bond. Now you can see that at the top, this growing protein chain. One amino acid at a time. Everything works by a code. Pretty cool stuff. You can see down the line, we got a stop codon coming up. Obviously, this process is about to stop. What ends up happening is some other protein that you're going to see as yellow is just going to enter the picture here in a minute. That's basically going to act like a pair of scissors. So that thing's going to come on in, act like a pair of scissors, and release your protein now that you just made. You're done. Who knows what that protein's going to go on to do? Maybe it's going to be forming part of your skin. Maybe it's part of your blood. Maybe it's part of your liver. Whatever. But what you know is you need to have it. So that's how it's made. Everything works by a code. So that's it with that. Bring out those other sheets I was talking about. The ones that Mrs. Hatcher's biology students are the best teacher or the best students kind of deal. Now that probably makes a lot more sense too. This sheet right here. I know you guys had a pretty easy time with this thing though. But now I want you to kind of see this is a good sheet to wrap up everything we talked about. No, that's fine. If you need whatever you need, I just totally...
Nope, you're good.
Okay.
Let me shut the
So when I
camera
take a
off.
picture, sorry. When I take a
Okay.
picture, it's upside down.
Oh, okay. So if you did the first one right, you have 3 different sets of codes here. If you did the first one right, your sentence should read, always drink upstream from the herd. You need to have every word there. Always drink upstream from the herd. But you do have a start in the beginning and a stop at the end. And in between there is your protein, basically, that you just made. Okay, the second one, Mrs. Hatcher's biology students are the best students. All right, and also though you have a start in the front and a stop at the end. The last one, I messed up on purpose. It should read, Wayne, Trace, raters are red, white, and laboratory. Okay. Why do you think I did that on purpose?
So we can automatically put blue.
Yeah, actually that's one reason, to prevent laziness. Okay, really, so because I've had quite a number of people who just put blue down there because that's what they think, huh?
That's what I did first.
Yeah,
Then I checked it.
But and so that was one reason, but that's actually not the main reason. It was actually kind of to prove a science point. What do you think? Think about like when something goes wrong.
Then I look mess up.
Yeah. The whole point of that was, if you have a mistake in your DNA, it's going to make the wrong RNA, which is going to bring in the wrong amino acid, which is going to make the wrong protein. Remember, guys, we're big bags of proteins. If our proteins aren't working right, you're in trouble. Remember that movie we saw at the very beginning of the year, the real sad movie with the kid with Tay-Sachs, the baby who didn't make it past a few years? It was the disorder where their brain, the fats build up in their brain and kind of squashes your nerve cells in your brain. That genetic disorder was caused by one letter of your DNA being wrong, which went on to code for the wrong RNA, which went on to code for the wrong protein. That protein doesn't work in that little boy. So what happened, in that particular case, the protein was supposed to help break down fats in the brain. Now, we all have that. We all have a working copy of that protein. But that particular person, that little baby, did not. We also saw another little clip with cystic fibrosis where there was another little kid who had to have his back padded out because mucus kept building up inside. Genetic disorder. That baby had something wrong with its DNA, made the wrong RNA, made the wrong protein, and in that case, the protein was supposed to work by moving salts across the plasma membrane. When you don't have the protein there doing its job, the salt contents are going to get all screwed up from the inside to the outside of the cell, and it causes mucus to build up. You and I, we have that protein, okay? All genetic, whenever you hear of something being a genetic disorder, that's how it all starts. You have a problem, you have something wrong with your DNA. Even if it's just one letter. Sometimes it's one letter, sometimes it's more. This is also why, moms, dads, grandmas, grandpas, doctors, everybody else tells you, don't smoke. Don't go to tanning beds. Don't lay out in the sun for all, hours and hours and get burnt. Don't lick pesticides. I don't know. All these things, okay? Stay away from chemicals. Stay away from anything like that because all of those things are bad because they will go in and mutate your DNA. That's the problem with them. And if they mutate your DNA, and if it happens in a portion of your DNA that's actually going to go on to code for a protein, you might be in trouble. Now, you'll luck out, guys. Keep this. You'll luck out if there's a mutation in your DNA that is in one of those introns. Okay? Remember, those introns kind of got sliced out. You're lucky. But if one of those mutations occurs in one of your exons, you're kind of not so lucky there. So, yeah, this is why they say to stay away from naughty, bad things and don't go to tanning beds and yada yada yada.
I tan all the time.
I know.
I don't. I'm not.
I don't know if people do that anymore. I know it was really popular back in.
You can tell.
Yeah, it was really, really popular years ago. I don't know if people still do it as much anymore.
I tan when
People tan during the summer.
People say, like, if you're going to go on vacation and you don't want to get burnt, I know there's some good, or if there's a wedding you're going to go to and whatever. But
I don't even put any tan lotion on. I just naturally tan.
It's not really the only way you can do it because you tan outside, you get tan lines. You use the lotion, you get orange skin that doesn't look real.
Yeah.
So it's either that or a chance of death.
Yeah. No, I know. A million things can kill you. I know. You're right.
Or you can be like me and
You're
just be
right.
natural tan.
So, yeah, that's it for today, people.